Welcome back, everyone. So we are on part 3 of our topic 3, the building blocks of life. So far we've been looking at the cell membrane and basically looking at how things enter and leave the cell. So now we're actually going to get into the biological molecules of life. So we're going to see what actually enters and leaves the cell. So we're going to look at the macromolecules of life. So by the end of this video, you should be able to describe the 4 major types of biological polymers, which is a fancy word for macromolecules. You should also be able to understand the functions of the 4 major types of polymers, as well as name the monomers of the 4 major types of polymers, which are the carbohydrates, lipids, proteins, and nucleic acids. And then you should be able to identify the atoms that make up the 4 major types of monomers. Okay, so we're going to delve into some chemistry here as to what makes up these macromolecules of life. So let's start out by looking at the biological polymers. So those 4 major macromolecules of life are as follows. So the carbohydrates, so think of sugars when you think of carbohydrates, the lipids, so think of fats, the proteins, and then the nucleic acids. So when you think of nucleic acids, think of like DNA, genetic information. So some of these you're very familiar with because they make up our diet, right? So our diet consists of a combination of carbs, lipids, and proteins, or carbs, fats, and proteins. And then everything has nucleic acids in it. So we're going to talk about these 4 macromolecules of life, which basically make up every part of your body. But before we get into the 4 major macromolecules, we need to dive into the major element that is part of all 4 of those macromolecules, the carbon atom. So we haven't really discussed the carbon atom in detail yet. So if we recall from back in topic 2, the carbon atom is, it's basically, going back, it is an atomic number of 6. So if we, did our little electron shell, if I drew that, okay, so carbon, its first shell would have 2 electrons, and then its second shell would have 4. Okay, so if it has 4 electrons, that means that it either needs to gain 4 or lose 4, which is highly unlikely. So it's going to be forming 4 covalent bonds. Okay, it's going to basically want to form covalent bonds, as many as 4 different atoms. So it's here's an example of one. We have methane. Methane is the simplest form, and it's just basically a carbon with 4 hydrogens attached. Okay, so carbon is the basis of anything that's organic, which you are organic. And so we're going to be talking a lot about carbon because all of our macromolecules are carbon or organic based. So let's start out with our first macromolecule, the carbohydrate. So carbohydrates, what do they do for you? Think about when you have food that are full of carbohydrates. Well, mainly they provide energy because this is our source of sugars. So think about foods that are high in carbohydrates. They tend to be high in sugar content, such as, like our sweets that we eat, the candies, the sugars, fruit, those products, fruit, breads, they have more of the complex carbohydrates, pasta, breads, things like that. They have stored sugar, so they are a good source of energy. So their job is to provide an energy source for our cells. They also provide structural support. So we'll talk about how they provide structural support as well. Now, as far as the molecule carbohydrates, the monomer, and we'll talk about the monomer in just a second. The monomer follows a ratio of 1 carbon to 2 hydrogens to 1 oxygen. So when we talk about the different subunits of carbohydrates, each of the subunits, they all have this same ratio where they have 1 carbon to 2 hydrogens to 1 oxygen. So for example, if we think of glucose as an example of a simple sugar, monosaccharide, if you want to talk about that, it has 6 carbons, 12 hydrogens, and 6 oxygens. So it follows that same ratio where we have twice as many hydrogens to carbon and oxygen. Okay, so it follows that ratio. And then we're going to talk about the 3 subtypes of carbohydrates. So we have the monosaccharides, the disaccharides, and the polysaccharides. So saccharides, just think sugar. Okay, so mono means like 1 sugar, di would be 2, so 2 sugars, and poly means many, so many sugars. Okay, so it's kind of easy to remember as long as you remember saccharide means sugar, and then mono 1, di 2, poly many. Okay, so we're going to go through each one of these subtypes and talk about them. Okay, so let's start out with the monosaccharides. So remember, mono means 1. Saccharide actually means sweet, so think sweet or sugar when you're thinking about monosaccharide. So these are considered the simple sugars. These are the monomers of our polymer, the carbohydrate or the polysaccharide. So here are some examples of our monosaccharides. Okay, so these are the subunits that make up the carbohydrate. And they're typically, as we said before, they all follow that same ratio of 1 carbon to 2 hydrogens to 1 oxygen. And typically our monosaccharides range from 3 to 6 carbons. So here's an example of 3 different monosaccharides. All of them have 6 carbons. So right away we all know that they have 6 carbons, 12 hydrogens, and 6 oxygens. Okay, and they all have, so they actually all have the same chemical formula, but they have different structures to them. So we can see some examples. So glucose, galactose, and fructose. And you can see that, there's just slight differences to them. Glucose and galactose, they're pretty similar. The only thing is that we have some switching of where the hydrogens and the hydroxyl groups are in the skeleton, the carbon skeleton, and then fructose where the double bond is. Okay. As I said before, the carbohydrates, their function is an energy source for our cells. So in topic 4, we're going to talk about energy production, and so we'll talk about ATP, cellular energy. And so glucose is very important in as a chemical energy source for our cells. Okay, so our cells actually convert glucose into ATP, or the energy stored in glucose to ATP through a process known as cellular respiration. So we're going to talk about that in topic 4. But for now, just realize that these monosaccharides, they are an important source of energy for our cell. And then, as you can tell just from looking at the 3 examples here, glucose, galactose, and fructose, they all end in the suffix "ose." Okay, so they're all oses. So that's, kind of a thing that if you see in your reading, the suffix "ose" for one of these terms most likely it's a monosaccharide. Okay, so that kind of gives you away. Okay, so not always, but sometimes you can kind of guess that it's going to be a monosaccharide. Okay, but not always. Like sucrose is a disaccharide, but you know, most likely. Okay, so here are the monosaccharides. So mono means one, saccharide means sweet or sugar. So this is considered the monomer of the polymer, the carbohydrate. So the subunit monomer of the polymer, the unit. So the next subunit or subtype of our carbohydrate is a disaccharide. So disaccharides are basically when we stick two monosaccharides together through a process known as dehydration synthesis. So let's look at that word dehydration. What does it sound like? Dehydration. Well, what happens when you dehydrate? Basically, you're dehydrated, you lose water, right? So that's exactly what happens during a dehydration synthesis reaction. So basically, we have two monosaccharides. Notice, look at the formula. They're exactly the same ratio. We have 1 carbon to 2 hydrogens to 1 oxygen. So in this case, we have a glucose and a fructose. They're going to be joined together to form sucrose. Sucrose is table sugar, okay? So whatever you use, like making cookies, that's sucrose. So that's just basically a disaccharide that's formed from glucose and fructose. So in order to form sucrose, it undergoes a dehydration synthesis reaction where we take glucose and fructose and water is removed, H2O. So in order to remove water, undergoes dehydration synthesis. So we have to remove a hydrogen and 2 oxygen, or excuse me, 2 hydrogens and 1 oxygen. So you can see here, if we count this all up, if we remove 2 hydrogens and 1 oxygen from this combination, we end up with this chemical formula, which is sucrose. Okay, so we still have 12 carbons, so they add up. Instead of 24 hydrogens added together, we lost 2 of them for the water molecule. And then instead of 12 oxygens, we lost 1 of the oxygens for the water molecule. So think dehydration, we remove a water molecule to bring the monosaccharides together. Okay, so basically we're taking this molecule and we're bringing them together. In the process, we're going to remove the 2 hydrogens. So we remove a hydrogen here and a hydroxyl here and stick them together. Okay, so they bridge together to form a sucrose molecule. So that is an example of a disaccharide. So two sweets or 2 sugars together, and they form a covalent bond together, so a very strong bond together. And then finally we get to our polysaccharide. So polysaccharide is our polymer. So polymer is just a long chain of monomers or monosaccharides in our case for our polysaccharide. And they're all linked together with covalent bonds. Now in our previous example, we've been looking at our, let me go backwards. You can see that the way that we're writing our monosaccharides, they're basically just written in these carbon skeletons. But in reality, these molecules, they are in a ringed structure. So if I show them, this is actually what they look like. So they're in these ringed structures. Okay. So when we have a polysaccharide, it's basically just a bunch of glucose molecules in this case, not the chitin, but the any of these cellulose or glycogen or starch. It's just a bunch of glucose molecules, in line, held together with covalent bonds. Okay. And they form different types of polysaccharides. So we have three types that form these polysaccharides that are made of glucose. Okay, so these are the examples here. So starch. So this is starch. Starch is basically just a bunch of glucose held together, linked together by covalent bonds. And if you think of starch, what food products are high in starch? So you can think of like potatoes, for example, pastas. So basically things that come from plants, right? You don't think of meat having a lot of starch in it, right? It doesn't have starch. So it comes from plants. It's actually a storage of sugar in plants. So plants, they store sugar away as starch. So that is a food storage for them. So think back, back in those, when we talked about the components of the cell, think of the vacuoles. So that's one place that they store excess sugar in the form of starch so that they can, put it away and then use it later on if they need to. Okay. We don't form starch, but we still need to store away our sugars. So we do that in the form of glycogen. So glycogen, it looks a little different. You can see how it's kind of kinked here. We have one coming off, a glucose chain coming off of a glucose chain. And so this is how we store our glucose. Okay, so it's still a polysaccharide, and it's still just linked together a bunch of monosaccharides or glucoses. So glycogen is the stored form of glucose in humans. Okay. And then finally we have cellulose. So cellulose is also made of glucose, but it's different. You can see starch is all one direction. Cellulose is opposite directions of the glucose. And this is what makes up the cell walls of plants. Okay, so cellulose, they have that rigid cell wall that is just outside of their cell membrane, which we talked about in the last video. And it's made up of cellulose. And you have eaten lots of cellulose whenever you eat vegetables or anything like that. And as far as our diet is concerned, it's considered dietary fiber. Okay, so we can't really digest it, and so it's the fiber that makes up our diet. Okay, so these are all made of glucose. It's just different ways the glucose is bonded. Okay, they're all formed covalent bonds, but just the way that it's situated, right? And then chitin is a little different. This is, it basically makes up the cell wall of fungi, but also makes up the exoskeleton of some of our arthropods. And if you notice, it's not glucose. Well, there is some, there's other things there. There's nitrogen. So, it does have glucose there, but it also has nitrogen. So that's why I didn't want to lump it in with these guys. So that's just another type of polysaccharide. Okay. So those were the, carbohydrates. So remember, carbohydrates, they provide energy source for our cells, but they also provide a structural support for the plant cells, okay, as well as some animals. Right. Next, we have our lipids. So lipids, I always think of fats, but there's other things that lipids do besides just are related to fats. So lipids, it's a very diverse category. It's not like the carbohydrates where they have a monomer and then you stick two monomers together, the monosaccharides together, and get a disaccharide, and then you get multiple monosaccharides together to form a polysaccharide. It's not so easy. Lipids, they are categorized based solely on that they are hydrophobic, okay, and that they are composed of non-polar molecules. That is it, okay? So that's their classification. Okay, so they look, they can have different structures, different, they look different from each other, but they are categorized because they are hydrophobic non-polar molecules, right? And they have a diversity of functions. So they are energy stores for us. So think about, fat in your body. So they can store energy for us that eventually we can, if we've used up our glycogen stores, we can also use fat that will burn as an energy source. They provide insulation. So, think about like polar bears or whales or, even us. We have layers of fat to provide for insulation. Also think of hormones. So hormones are, some of them, not all of them, in our bodies are lipid based. Okay, so like our estrogen, testosterone, those are examples of lipid-based hormones. And they also, remember that phospholipid bilayer. So our cell membranes are composed of lipids. Okay. So here's some examples of things that are lipids. So of course those fats, so think the fatty acids, which make up the tails of our plasma membranes. Oils, so when you think of when you're cooking, you have vegetable oils. Those, of course, are made of lipids. Waxes, okay, so waxes, think of like what's on the fur of our river otter that keeps the river otter warm. So the fur has like this waxy coat that is hydrophobic, that protects the river otter from the elements, keeps them nice and warm. Phospholipids we talked about, and steroids, those are examples of lipid-based hormones. Okay. So we're going to talk about like kind of some different types and types of lipids. So an example of a type of lipid is a fat molecule, okay? So if we look at, let's look over at a fatty acid, okay? So if we look at the structure of the fatty acid, this is a saturated fatty acid, by the way. It basically is just carbons and hydrogens with an oxygen at the end. Okay, so it's basically a bunch of hydrocarbons is what we call it. Okay, so when we have a fat, it is composed of a glycerol and fatty acids. So we have, in this case, a triglyceride consists of 3 fatty acids, 1, 2, 3, with a glycerol. And in this case, you can see that it's basically just a bunch of carbons and hydrogens. Okay, so basically hydrocarbons everywhere, and just a few oxygens spread in there. Okay. Now, some of you, you might be looking carefully at food labels and taking a close look at saturated versus unsaturated fats, but you might not know what the difference is. So here is an example of a saturated versus an unsaturated fat. Now, if you look carefully, can you see the difference? So the saturated fat looks pretty linear, right? The unsaturated fat has a kink, okay? So that kink is caused by a double bond. So basically what happens here is that the double bond means that the unsaturated fat has one less hydrogen in this case, or it can have multiple double bonds throughout it. So it's not as saturated with hydrogens as a saturated fat. And because it has these little kinks in the chain, it's more spread out. And so if you think about food products that have more, that are more unsaturated fats, they tend to be more liquid at room temperature. So think of like vegetable oils, like olive oil is, liquid at room temperature. So plant-based products tend to be higher in unsaturated fats. Whereas like butter, animal-based products tend to be higher in saturated fats. So they're saturated with hydrogens. So each one of their carbons is saturated with hydrogens. So unsaturated, think, there's less hydrogens, and so it leaves little kinks or openings, makes it more spread out. So liquid at room temperature, saturated, very saturated with hydrogens, so there's no kinks in the chain, and so it's going to be solid at room temperature. Okay, so that's what the difference is. All right. Now, you may have heard about trans fat, and we know that's a bad word, right? So we want lately. We hear that we want to avoid trans fats, and there's even been, I think there was a measure passed a while ago that we're phasing out trans fats from our food products. And so what is a trans fat? Well, first of all, a trans fat is a form of an unsaturated fat with hydrogen atoms that basically have a different bonding. So here is an unsaturated fat that, looks pretty normal. Okay, so here's the little kink. Okay, and remember the kink is caused by that double bond. Okay, and cis just means that the hydrogens are on the same side. But the food industry, they like to, make things cheaper, but taste the same, right, as the more expensive product. And so, there's a big push, way back in like the 70s to take something like, butter that tastes really good and, make a cheaper product that looks like butter, but is not actually butter. Okay, so think of margarine. So what is margarine? Margarine is not butter, but it looks like butter, right? But it's made from vegetable oil. Okay, so what happened is they did a hydrogenation process. So basically they took an unsaturated fat, vegetable oil, and they hydrogenated it to make it solid at room temperature. And so to do that, they ended up creating a trans fat molecule. Okay, so they kind of took this unsaturated fat and they tried to stabilize it to remove the kink to make it solid at room temperature. And in doing so, this is what ended up happening. So it kind of stabilized it. But we're finding out that it caused some health issues because our bodies don't really know what this is. It's not exactly a saturated fat that our body's used to recognizing, but it's not exactly an unsaturated fat that our body recognizes. So our bodies don't really know how to process them the same ways as a true fatty acid. And so that's why we're having it's contributing to health issues, which is why we're phasing it out of, there's laws that have been put in place to kind of phase out these trans fats from our diet. And it's basically, you don't have to worry about trans fats in healthy foods, right? It's basically in heavily processed foods. So you want to avoid things like, margarine, shortening. Some peanut butters still have them, but they're being phased out. Okay. All right, and then finally we're going to get into the phospholipids and the steroids. So we've already talked about phospholipids, so they're the main component of our plasma membranes. And remember those phospholipids, they have a phosphate head and then the fatty acid tail. or the lipid tail. So you can see the lipid tail, those are made of fatty acids. And you can see that the fatty acids, they can be, the kind of saturated fats, fatty acids, and as well as unsaturated fat. And so they're linked to our phosphate head, which is hydrophilic, and then the lipid part being hydrophobic. And then finally we get to our steroids. So steroids are kind of interesting because they're not, linked together like all of the fatty acids we've been talking about. They're in a ring structure. Okay? So they have a ring structure. And steroids, remember in our cell membrane talk or plasma membrane talk, we have cholesterol that actually is part of the cell membrane. So it helps stabilize the cell membrane, so it's not completely, even though the cell membrane is fluid, it's not completely so fluid that it's flying everywhere and into chaos. It helps stabilize it. So we do need cholesterol in order to stabilize our plasma membranes. Other steroids could be like our sex hormones, estrogen and testosterone. They have this kind of same ring structure shape as our cholesterol. Actually, cholesterol is the base. So testosterone and estrogen, looks very similar to this molecule. Right, so we're going to watch a video about.
Contrary to popular belief, not all fats cause heart diseases and are bad. In fact, most fats, in adequate amounts, are required for normal bodily functions, especially brain functions. There are also good fats that actually decrease the risks for cardiovascular diseases. A fat molecule is composed of a glycerol head and 3 fatty acid tails, each of which is a long hydrocarbon chain, a carbon skeleton bound to hydrogen atoms. When all the carbons are fully bound to hydrogens, the fatty acid is said to be saturated. All the bonds between carbon atoms are single, and the hydrocarbon chain has a straight shape. A fat molecule made entirely of saturated fatty acids is a saturated fat. Due to their straight tails, saturated fats are compact and solid at room temperature. On the other hand, when the hydrocarbon chain has fewer hydrogens, it is said to be unsaturated. Instead of binding to a maximum number of hydrogens, some carbon atoms bind to each other via a double bond. The presence of double bonds may bend the hydrocarbon chain, creating gaps between molecules, making them less compact. As a result, unsaturated fats are usually liquid at room temperature. A fat molecule that contains only one double bond is a monounsaturated fat, while one that has multiple double bonds is polyunsaturated. Dietary fats provide fatty acids for the synthesis of the cell membrane, a vital component of all animal cells. The gaps in unsaturated fatty acids provide membrane fluidity, facilitating membrane transport and cellular signaling. While both types of fats are needed for an optimal composition of the cell membrane, too much saturated fat, which is commonly the case in a typical American diet, would make the membrane rigid and hinder cellular responsiveness. Membrane fluidity is most important in the nervous system, where neuronal response requires extremely fast cellular communication. A certain ratio of unsaturated to saturated fatty acids is also required for the formation of myelin, the insulating material that wraps around axons of neurons and speeds up the conduction of electrical signals. The body is capable of synthesizing all the fatty acids it needs, with the exception of polyunsaturated fatty acids omega-3 and omega-6, which must be obtained from the diet. These are known as essential fatty acids. In general, unsaturated fats are healthier than saturated fats. Unsaturated fats decrease the risks for heart disease by reducing the amount of bad cholesterol, LDL, and increasing the good cholesterol, HDL, while saturated fats increase both good and bad cholesterol. But not all unsaturated fats are equal. In fact, a type of unsaturated fat, known as trans fat, is the unhealthiest of all. A double bond can give rise to 2 possible configurations, cis and trans. Cis is when the two hydrogen atoms are on the same side of the bond, while trans is when they are on the opposite sides. A cis double bond bends the fatty acid molecule, while the somewhat more symmetric trans configuration does not. A trans fat is therefore similar in structure to a saturated fat. More importantly, trans fats rarely occur in nature, so the body does not have the necessary enzymes to break them down. Diets rich in trans fats increase the bad cholesterol LDL and reduce the good cholesterol HDL, having the most detrimental effect on blood vessels. Trans fats are found mainly in partially hydrogenated oil products, such as margarine. Because unsaturated fats are less stable and spoil faster, food manufacturers add hydrogens to make them more saturated through a process known as partial hydrogenation. This process not only prolongs shelf life of vegetable oils, but also turns them into solid or semi-solid products, which are preferred by commercial bakers for their low cost and wide range of different textures. Unfortunately, partial hydrogenation also converts some of the cis double bonds into trans configuration, producing trans fats. The FDA has officially banned production of partially hydrogenated oils in June 2018, but products made earlier may still be in use until January 2020. Thank you for watching. If you like our videos, be sure to...
Alright, so that video gave not only a review of the different types of fats, but also, so a little bit of health. Think about the cholesterol issues. So you want low LDLs and high HDLs, and which fats are better for your diet. So let's get into the third type of macromolecule of life, the protein. So I always think of proteins as the building blocks of life, because basically everything in your body is, when you look at everything that you're looking at, when you look at your body, is protein based. So protein is composed of one or more chains of amino acids. So amino acids are the monomers of the polymer protein. And proteins carry a lot of functions in our body. So think of structural, so they make up the structure of your body. So muscles, the fibers, everything that is constructed within your body. Regulatory, so heart contractions, all of that. Contractile, so again, muscle contractions. Protective, immune response, okay, would be a function. Transport, so transporting things from one side of the body to another, so like transport vesicles. Storage, storage in the form of, think of a chicken egg. The egg white is made of albumin, which is an energy storage for the developing embryo of a chicken. Enzymes, so those are catalysts that provide the energy to drive a reaction. They're pretty much all protein. And then lipids can be, hormones can be lipid based, but they can also be protein based, like insulin is a hormone that is protein based. So let's talk about the monomers of proteins, the amino acids. So when we take a look at an amino acid, I'm going to show you way up here. There is a carbon in the center, and then remember carbons, they can have up to 4 covalent bonds. So one of the covalent bonds is with a hydrogen, one is with an amino group, okay, so that amino group, amino because there's a nitrogen, and then the other is with a carboxyl group. Okay, so we'll just talk about the amino group, just think of nitrogen, and then the carboxyl group, think it's got COOH. So the amino, and then carboxyl means like an acid, so amino acid. Sorry, it's kind of strange that if we really went into depth about like the chemistry, carboxyl is an acid. So amino acid. So one side is the amino group, and then the other side is the acid group. And then finally we have at the bottom, we have the R group. So the R group is different based on the type of amino acid. So everything on top here is the same for every amino acid. So you have an amino group, a hydrogen, and a carboxyl group for every type of amino acid. The only thing that's different is the R group. Okay, so you can see that alanine has, basically this is called a methyl attached for their R group. Valine has, something else. Basically, it's a little bit more like 3 methyls and a CH. For valine, lysine has something else. Okay, aspartic acid, hey acid, there's the acid. Okay, they actually have, something else. So what determines the name of the amino acid is the R group. Okay, otherwise everything on top is the same. Okay, and that's important because when we talk about the bonding of the amino acids, when we take 2 amino acids and we connect them, they're actually going to be connected from the amino group to the carboxyl group of a nearby amino acid, and they're going to form a covalent bond. And so when we have amino acids linked together, they're going to form a polypeptide, which if we have polypeptides, we form a protein. Okay, so polypeptides are the polymers of the amino acids. Okay. So when we talk about how a protein is formed, it's actually pretty complex how they form. So we start out with the primary protein structure, which is just basically those amino acids linked together. And then remember, if I go back, the way they are linked together is basically we have an amino group from one amino acid attached to the carboxyl group of a nearby amino acid. So they're just linked together in a row. Okay, so just imagine, this carboxyl group is going to be attached to another, an amino group of a nearby amino acid. Okay, this amino group is going to be attached to a carboxyl group of a nearby amino acid. So they're all going to be linked together. So that means all of their R groups are kind of facing, that kind of direction. So here, that is a link chain of amino acids, and they're all held together by covalent bonds. Actually, those covalent bonds are called peptide bonds. It's just a name for a covalent bond because it's a protein, peptide. And so those are the amino acids linked together. So that's the primary structure. But then, because we have if we go back, this H involved, this H is going to start forming hydrogen bonds with nearby amino acids. And so we're going to start seeing a folding occur. So this folding is going to cause either a beta pleated sheet or kind of an alpha helix form. So imagine this chain is going to start folding, and it's due to the hydrogen part, of our amino acid. Okay, so the hydrogen is going to start, interacting with nearby amino acids. So, there's a little hydrogen group, and it's going to, hey, I'm, interested in maybe, some bonding going on, hydrogen bonding nearby. And so they're going to start folding into each other. And as they fold, they're going to form these kind of secondary structures, either an alpha helix or a beta pleated sheet. Okay. Next comes the R groups. Okay, so let me go back again. These R groups are going to start coming into play. Now each one of these R groups has a different function. Some of them are acidic. Okay, if they're acidic, that means they're water soluble, right? Some of them are hydrophobic. that means that they don't want anything to do with water. Okay, so there's different properties to these R groups. Okay, so based on their properties, will determine how they react with each other. And so they're going to start folding based on those R side chains. So for example, if that R group is hydrophobic, it's not going to want to be hanging out on the outside here. So it's going to be wanting to fold inward, okay, towards the middle to be protected from the water, the liquid part. So you're going to see that instead of kind of a two-dimensional shape, it's going to start folding into a three-dimensional shape based on the properties of those R side chains. Now, when we get to this tertiary protein structure, this might be it. This might be the protein. However, sometimes we can have a quaternary protein structure form. So a quaternary protein structure is when we have multiple polypeptide chains. So that means we have multiple of these guys, okay? So we end up with more than one of these together, okay? So we end up with more than one amino acid chain, and that forms a quaternary protein structure. All right, so it's pretty complex, right? When we get to these proteins, they look pretty complex. Actually, they look kind of like a blob, but it's a complex blob with a lot of features involved. Now, because it's so complex, it can be affected by the environment. So we can have our proteins, they can become denatured if they're exposed to changes in the environment. So if there is a change in temperature, they can become denatured. So for example, if the temperature increases too much, basically the tertiary structure can start to unfold. And if it's unfolding, then the protein no longer functions correctly. Or if the pH changes, then the protein can unfold. And if it's unfold, then it's not going to function properly. Or if it's exposed to different chemicals, then it can unfold. And if it's unfolded, it can't function properly. So there's a number of environmental conditions or changes that can occur that causes the protein to denature. And if denaturation occurs, then the protein loses its function. So loss of shape loses the loss of function, okay? So that's why we have a lot of issues with proteins. If, for example, think about if you have a fever, okay? Now, usually, if you have a fever of like 100, no big deal, you just rest. But what happens when your fever spikes to like 106? It's emergency room time, right? So when your temperature gets really high, it's emergency room time because a lot of your enzymes, which are protein-based, they could start to denature. And if they're denature, they lose their function because they're unfolding. And that can cause a lot of problems. So that's why, kids, when they have a temperature of usually like 104 or above, pediatricians recommend the kid, go to the emergency room right away because we got to figure out, how to bring that temperature down. So that they don't lose, they don't have their proteins denature in their bodies. Okay? So denaturation. All right, so let's go to the final of our macromolecules of life, the nucleic acids. So nucleic acids are the holders of genetic information. Okay, so they're the DNA and RNA. So DNA, if we break that word down or acronym down, it stands for deoxyribonucleic acid. So deoxyribonucleic acid. Whereas RNA, it's a little shorter, ribonucleic acid. So they're nucleic acids. The only difference is deoxyribo and ribo. So what does that mean? Well, nucleic acids, that's the macromolecule. So the monomer, or the subunit, are the nucleotides. So what is a nucleotide? A nucleotide is made of 3 components. We have the phosphate group, we have the sugar group, and we have the nitrogenous base. Okay? And this is how it's situated. Okay? So we have the phosphate group is attached to the sugar, and then the sugar is attached to the nitrogenous base. Okay? And as far as the sugar is a pentose sugar, which pentose means 5, so it's a 5 carbon sugar. Okay? And we'll get into more detail. So basically that deoxyribose is a type of pentose sugar. Ribose is a type of pentose sugar. So it's saying deoxyribose sugar makes up DNA, ribose sugar makes up RNA. So let's talk about DNA first, and then we'll get into RNA. So DNA, actually I don't even know if we get into RNA. So let's talk about DNA. DNA is 2 strands of the DNA molecule, and it's wound together in a double helix. Okay, so we have 2 strands of these nucleotides, these subunits, and they're held in place by basically a bond. So if we look at each strand, the strand itself, one strand is made up of a backbone, and that backbone is made up of the bond between one nucleotide's sugar and another nucleotide's phosphate. So we call this backbone the sugar phosphate backbone. Okay, so it's almost like a ladder structure to me. Okay, so the what holds the ladder up is the sugar phosphate backbone. And then the rungs of the ladder, the steps of the ladder, are basically the nitrogenous bases. Okay, so we have one nitrogenous base of one DNA strand, forms a hydrogen bond with a nearby a nitrogenous base of the opposite strand, the other strand. Okay, so there's a hydrogen bond that holds them together. And remember, hydrogen bonds are weak bonds, so this is kind of a temporary bond. But the sugar of one nucleotide and the phosphate of a nearby nucleotide, they're held together by a covalent bond. And remember, covalent bonds are strong. So the sugar phosphate backbone is strong. The hydrogen bonds are weak. So the nitrogenous bases, they're held together by a weak hydrogen bond. So before we get to the review questions, I want to show you this Amoeba Sisters video. So if anybody has heard of the Amoeba Sisters videos, they are great study tools. If you haven't looked them up, they're in YouTube, and they go over the major biology concepts. So if you're ever struggling with a biology concept, just look them up, and they have some cute videos that are very informative and help you remember certain things about the concept. So let's go ahead and watch this video on macromolecules.
I want you to think for a moment about your favorite food. What is it? We all have different food preferences, but food is a source of large molecules that are needed for life, called biomolecules, also known as macromolecules: carbohydrates, lipids, proteins, and nucleic acids. In different types of food, you might find higher amounts of these biomolecules. For example, peanut butter can be high in both proteins and lipids. And while we'll mention examples of foods that contain different biomolecules in this video, this is not a video on nutrition. This is a video on the classes of biomolecules and their importance to the structure and function of cells, and therefore also to the entire organism, because organisms consist of these cells. Before we get into details about the 4 biomolecules, we need to talk about one very important vocabulary word: the word monomer. A monomer is a building block. If I had some large substance, the parts that make up that substance are called monomers. With each biomolecule class, we will mention its monomer if it applies. We'll start with the biomolecule class of carbohydrates. Generally, food sources such as bread, pasta, fruit, and vegetables tend to be high in carbohydrates. The monomer of a carbohydrate, remember that means the building block, is the monosaccharide. One example of a monosaccharide is the sugar glucose. Glucose is a pretty big deal considering it's used in cellular respiration to make ATP, an energy currency. If two monosaccharides, such as glucose, are combined, you get a disaccharide. For example, put these two glucose sugars together and you get the disaccharide maltose. By the way, a lot of sugars have that OSE ending. Glucose, maltose, lactose, sucrose, fructose, I could go on. The ending OSE can be a carbohydrate clue. If you join a lot of monosaccharides together, that can combine to make a polysaccharide, a big carbohydrate. So how are carbohydrates important to an organism and the structure and function of its cells? Well, plants have cell walls of carbohydrates, specifically a large one known as cellulose. Fungi have cell walls of carbohydrates too, specifically one known as chitin. Fun fact that carbohydrate chitin is also what makes up the exoskeleton of insects. We mentioned glucose is used to make ATP. That makes carbohydrates a fast source of energy for cells. So that's a big function for carbs, major energy source that cells can access. That energy can be stored in a polysaccharide form. Remember, polysaccharide is a big carbohydrate, such as starch for plants or glycogen for animals. Next up, lipids. Lipids include fats and oils. Examples of food sources could include butter or olive oil. This is a seriously diverse group, meaning there are a lot of types of lipids. There are triglycerides, there are phospholipids, there are steroids. These are just some examples. Most, but not all, lipids have building blocks of glycerol and fatty acids. One thing though that lipids generally share, generally they're hydrophobic, or at least contain a significant hydrophobic component. That means lipids, or at least a major component of them, don't like water. Lipids generally won't dissolve in water. And that's interesting, as generally the other biomolecules will. So how are lipids important to an organism in the structure and function of its cells? Remember how we said all cells, plant cells, animal cells, all cells, have membranes? Well, thank you phospholipids for that phospholipid bilayer, because that's right, lipids make up cell membrane structure. Lipids have a major function of being a source of long-term energy storage. Remember how we said carbohydrates were a fast source of energy, but when those carbs are used up, lipids have a function as long-term energy storage that an organism can use. Lipids are also very important for different types of insulation. The insulation may involve helping a cell carry an electrical impulse. For example, this myelin sheath made of lipids that can be found on many neurons. Or the insulation may involve temperature regulation, so thermal insulation. For example, the blubber on this adult harp seal is made of lipids, helping it conserve heat. Another cool function about lipids, many lipids act as hormones, which are chemical messengers for many different processes. Next up, proteins. Protein food sources include beans, meat, nuts, and eggs. The monomer of a protein is an amino acid. Usually proteins consist of many amino acids bonded together. There are different types of amino acids. So how are proteins important to an organism in the structure and function of its cells? Structure-wise, tissue like muscle tissue consists heavily of proteins. Proteins can make up all kinds of other structures, hair or collagen, for example. Proteins can also be found embedded in cell membranes as protein channels. And channels are important to that cell membrane structure with functions of letting certain substances pass through. Proteins can also be receptors, which are critical in cell signaling, so cells can coordinate actions together. Most enzymes are made of proteins. Remember, enzymes can build up or break down substances in all kinds of metabolic processes. Antibodies, which are made by immune cells in your body, are proteins, and they are involved in protecting your body. And some critical hormones, such as insulin, are proteins. And when we start talking about genes, genes are made of DNA, but many genes do code for proteins. And that brings us to our last one that includes DNA, nucleic acids. Nucleic acids include DNA and RNA, which we'll get to in more depth when we get to genetics. Nucleic acids have a monomer called a nucleotide. That's going to be an easy one for you to remember because nucleotide sounds like nucleic acid. By the way, you might wonder, are nucleic acids like DNA and RNA found in my food? And the answer would be yes. Whenever you eat something that came from something living, it actually could still have DNA inside it. For example, when you eat a strawberry, you're actually crunching all the cells that made up that strawberry, and in the nucleus of all those strawberry cells is DNA. Any type of life, like plants or animals or fungi or bacteria, they must contain nucleic acids like DNA to direct the cell's activities. How are nucleic acids important for your cells? Well, most of your cells contain all of your genetic information in the form of nucleic acids, DNA and RNA, and this is needed for the coding of your traits. Traits involved in structure and various functions. Okay, so we just did a very general tour of 4 biomolecules. Before we go, I just have to marvel at biomolecule structure, because it's absolutely beautiful. So if I arrange them in the order we did, carbs, lipids, proteins, and nucleic acids, how might we remember the major and most common elements in them? There's a popular mnemonic known as CHO, CHO, CHOM, CHOMP, although it's CHOMP with an N, to help me remember the C for carbon, H for hydrogen, O for oxygen, N for nitrogen, and P for phosphorus. Does that mean you won't find other elements in the biomolecules? Of course not, but it's important to understand the arrangement of these elements in biomolecules because the structure of the arrangement greatly impacts their function. You can learn more in our further reading suggestions in the description. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so again, I liked showing this video just because it gives you not only just a summary of the monomers and the functions of each of the 4 macromolecules, but it does give you those elements at the end with a good acronym to help you remember. So let's do the review questions. An example of a monosaccharide is A, fructose, B, glucose, C, galactose, or D, all of the above. If you chose all of the above, I would agree with you. So all 3 of these are examples of monosaccharides. So remember, all of these, they actually the same chemical formula. They have 6 carbons, 12 hydrogens, and 6 oxygens, just in a different kind of structural format. And when we put together the fructose and the glucose, we combine them in a dehydration synthesis reaction, we get sucrose, a disaccharide. Here's another question. Cellulose and starch are examples of, are they monosaccharides, disaccharides, lipids, or polysaccharides? If you chose polysaccharides, I would agree with you. So remember, polysaccharides are many sugars, so many, in this case, glucoses stuck together with covalent bonds. And cellulose, that is what makes up the cell walls of our plant cells. And then the starch is a glucose storage within, in this case, the potato plant. Okay. Here's another review question. Phospholipids are important components of, is it the plasma membrane of animal cells, the ring structure of steroids, the waxy covering of leaves, or the double bond in hydrocarbon chains? If you chose the plasma membrane of animal cells, I would agree with you. Well, and plant cells, right? Of any cell. Phospholipids, here we have our phospholipid that makes up our plasma membrane. And then we have the monomers that make up proteins are they nucleotides, disaccharides, amino acids, or chaperones? If you chose amino acids, I would agree with you. And sometimes you see this as peptides also, but hopefully most of the time you'll see them as amino acids. I think the workbook usually only calls them amino acids, but occasionally you might see peptides out there. So amino acids are the monomers that make up the proteins, or polypeptides, sometimes you'll see that. All right, that's it for part 3 of our topic 3, the building blocks of life. So now you should be able to answer the learning objectives. You should be able to describe the 4 major types of biological polymers, our 4 macromolecules, as well as understand their functions. You should be able to name the monomers of the carbohydrates. So that was the monosaccharides. Lipids, there wasn't one, okay, specific one, but you should be able to give examples of types of lipids. Proteins, what was the monomer? That would be the amino acid. And then nucleic acids, what was the monomer? The nucleotides. You should be able to identify the atoms that make up the 4 major types of monomers. Okay, so what made up a carbohydrate? So think of, the ratio I talked about. Lipids, think about those fatty acid chains. What were they made of? Proteins, think about that structure I showed you. And then the nucleic acids. Okay, look at that nucleotide I showed you. So you should be able to do all of that. And then finally, for the last video of topic 3, we're going to learn about how we synthesize these macromolecules.